everyone, and welcome to December MRM Highlights. My name is Jessica from Minnesota, and today I'll be talking to Lorenzo, Jérôme, and Matthias about their latest paper on a user-friendly cardiac protocol that acquires 5D whole heart images with just the push of a button. So let's meet the authors first.
I'm Jérôme. This is Lorenzo.
Hello. And I'm Matthias.
Nice to meet you. Okay, so will you guys start with a little bit of background about yourselves?
Well, I've started my PhD more or less 3.5 years ago here at here in Lausanne at the University Hospital of Lausanne with Matthias and Jérôme. And I've been working during my PhD mainly on projects related to the free running framework, so on cardiac imaging, and in particular with a focus on the extraction of physiological information, physiological motion information through different techniques.
Okay, hi, my name is Matthias. I did my PhD so long ago I don't even remember. I think it was 1996. I did that, guess what, in cardiovascular imaging at the ETH in Zurich. Then I moved to Boston, did cardiovascular MRI. Then I became a faculty at Johns Hopkins and did, guess what, cardiovascular MRI. And then 10 years ago, I moved to Lausanne, and guess what, the rest is history. I was also the president of the SCMR, the largest international society dedicated to imaging of the heart.
I'm Jérôme Yerly, and I finished my PhD in 2013. I was in Calgary with Dr. Richard Frame. I was mainly working on neuroimaging. And then I moved to Switzerland, July 2013, and I started working with Matthias, and I started doing cardiac imaging, basically. And I focus mainly on image reconstruction.
What drew you to doing cardiac imaging, like your whole career, and maybe you guys too, what drew you to switch over from neuro and get into cardiac imaging?
Well, I'm an electrical engineer by training, so I was always attracted to challenging technical problems. And at the time, it seemed to me as if the addition of motion is an additional problem to MRI, which I felt very passionate about. And I felt very fortunate to be able to include it in that research team at the time in Zurich and to work on that particular topic. And you know, it has never left me. I still feel very passionate about how to solve these motion problems, and that's why the research focus of my team is still there.
Well, for me, I'm an engineer by training as well, and I was doing neuroimaging during my PhD. But every time I used to go to conferences and I saw all those cardiac presentations, my engineer side was kind of very interested about all this motion and how we dealt with motion and all the reconstruction part of it. And basically, when I met Matthias and his group back in 2012, I think, at the SMRA in Utrecht, I got very interested about the project. So when he offered me a position here in Switzerland, I could only say yes.
Okay, so this one might be a little bit of a silly question, but what does free running mean?
So free running, when we talk about free running framework, is a different approach to cardiac imaging that we have kind of introduced. And the idea behind the word free running is that instead of looking for specific windows of acquisition, for instance, during the cardiac cycle and during the respiratory motion, in which we hope to have a static condition of the heart so that we can acquire an image that is not blurred. Instead, the free running approach aims to acquire continuously, so making the acquisition much more easy from the point of view of all the planning of the scan, and just dealing with the motion in a retrospective step, so after the acquisition. And this way also increasing the time efficiency of our acquisition and including much more information in our images.
Maybe I might add, for people who are imaging the brain, it's a no-brainer, so to speak, that pulses are played out continuously without interruption. For cardiac guys like myself, this is unheard of because you trigger the acquisition to the rhythm of the heart, as Lorenzo just explained. Now, being able to continuously acquire data, again, that was just not conceivable at the time, but now with modern tools that came along, all of a sudden it became possible. And if you think about it, I don't think there is any MRI sequence in the world that collects more data per unit time of the heart than the sequence we are thus discussing here right now. We started with this whole project not knowing that there will be new technology coming in that will be so critical in helping us, kind of to fulfill the promises and to complete the aims as we had written them in grant proposals. So I also find this is very fascinating that there are always new things coming in which you cannot really plan for. It's like young talent, like these two gentlemen sitting to my left, who come up with new ideas, new approaches, etc., which then all of a sudden open doors and remove hurdles. So this is, I think, this is super fascinating.
Before we go to the audio slides, can you just give me a brief overview of the impact of your work?
Perhaps, you mentioned the target audience before, that it's the general, the general person who is interested in MR. And if you look at imaging of the heart, everybody who does it tells you it takes forever, it's complicated to do, you need highly educated technicians. And I think we try to address all of the above. Anybody can find the heart and press the button because you only have to click one button. And then all the analysis, the extraction of the information is done retrospectively. So imagine a young patient with, congenital heart disease, where the plumbing of the vessels of the heart is not correct. So if the person who operates the scanner misses the correct scan plane in that case, and the cardiologist comes down to the MR suite and looks at these images and finds out, oh, there is a missed plane, then the proverbial horse is out of the barn, right? You would have to call the patient back to do a new investigation. In the case of our technique, not only is it simple and time efficient, but you have all the information in there. If you miss a plane, it don't matter. You just go in there retrospectively and find that plane. You will find that anomaly you're looking for, no problem.
Hello, I'm Lorenzo Di Sopra, and on behalf of my co-authors Davide Piccini, Simone Coppo, Matthias Stuber, and Jérôme Yerly, I'm presenting an automated approach to fully self-gated, free-running cardiac and respiratory motion-resolved 5D whole heart MRI. This study was conducted at the Lausanne University Hospital. Conventional cardiac MRI in current clinical settings suffers from limited ease of use and operator dependency, since many user interactions from highly qualified operators are needed to plan the acquisition. These interactions include cardiac resting phase selection, respiratory navigator positioning, complicated view planning, and many other manual settings selection. As a fun fact, we asked our technician Ledoux to count the number of mouse clicks that were necessary for a brain and a cardiac scan. Only 20 clicks were sufficient for a brain scan, while the cardiac one required up to 350 clicks. While these results are not very rigorous, they clearly indicate that the current clinical workflow for cardiac MRI needs to be simplified. Most of these user interactions are necessary to suppress cardiac and respiratory motion. For instance, cardiac motion is typically dealt with by ECG triggering, while respiratory motion requires breath holding or respiratory navigators. So the overall goal of this study was to simplify the workflow by developing a more time-efficient one-click approach to cardiac MR imaging. To achieve this goal, we introduced the free-running framework based on three fundamental pillars: a free-running whole heart 3D radial sequence, a self-gating physiology signal extraction, and a comprehensive motion-resolved reconstruction. The purpose of this study was to develop an automated, optimized and fully self-gated free-running framework for motion-resolved 5D comprehensive whole heart MRI, in which external gating or triggering devices are no longer needed. Here you have a schematic illustration of the whole study design. Let's now have a closer look to the different steps. Data acquisition was performed on 12 healthy volunteers with a free-running BSSFP sequence. ECG was recorded only for comparison purposes. The trajectory is 3D radial, and the first readout of each spiral was acquired in the superior-inferior direction for motion self-gating. The second step was the extraction of self-gating motion signals. 3 different methods for cardiac motion estimation were tested. K0 modulation using only the K-space central coefficients, principal component analysis, and independent component analysis performed on the Fourier transformed SI projections. Respiratory motion was also extracted with a self-gating PCA approach. To improve the extraction of self-gating signals, we implemented a correction that I'm going to describe in the next slide. The trajectory-dependent frequency component filtering is a procedure used to suppress the modulation induced by eddy currents on the self-gating signals. These trajectory-dependent modulations can be seen here as low-frequency oscillations when the signal is reordered according to the readout angle. To remove these modulations, the reordered signal is high-pass filtered and then sorted back in the temporal domain. The signal is now cleaned from the trajectory-dependent modulations, and only the modulations induced by cardiac and respiratory motions remain. Among the 3 strategies for cardiac signal extraction, the self-gating method that produced the smallest standard deviation relative to the reference ECG intervals was selected. The next step of the pipeline was data binning. The continuously acquired data were partitioned along both the cardiac and respiratory dimensions, using the estimated self-gating signals. For comparison, a reference cardiac binning was also performed using the ECG triggers instead of the self-gating triggers. 5D comprehensive sensing reconstruction was performed twice for each dataset, first using the self-gating triggers, then using the reference ECG triggers. Finally, the length and sharpness of coronary vessels were compared in corresponding reconstructed images. Now let's move to the results. I will start by presenting results about the signal extraction step and more specifically about trajectory correction. In this example, we can observe the effect of trajectory-dependent frequency component filtering on the spectrum of a self-gating signal. The trajectory correction separates the physiologic motion components in blue from the unwanted trajectory-dependent components in red, which otherwise would overlap in the same frequency range. The following results pertain to the precision of cardiac self-gating signals. Among the 3 different self-gating cardiac approaches, the PCA-derived triggers yielded the smallest deviation from the reference ECG triggers. The next results will show comprehensive 5D reconstructed images. In this example, the same dataset was reconstructed with the ECG-gated and the fully self-gated approach, with no visible difference in the resolution of cardiac or respiratory motion. Finally, we show the results of the coronary conspicuity measured from the reconstructed images. In these figures, we can observe the equivalent visibility of reformatted coronary arteries in the ECG-gated and self-gated images during both mid-diastole and systole. In the second subject, the same equivalence is shown for both the RCA and LID. These results are corroborated by the following objective quantitative measures. Looking at the measured parameters for both RCA and LID, for both length and sharpness measurements, for both systolic and mid-diastolic cardiac phase, no significant difference was found in corresponding ECG-gated and self-gated coronary images. We come now to the conclusions of this study. We have demonstrated that the self-gating triggers were precise relative to the ECG. This led to an equivalent objective image quality between the fully self-gated and the reference ECG-gated 5D reconstructions. Therefore, we can conclude that we have successfully developed an automated and fully self-gated free-running framework for motion-resolved 5D comprehensive sensing whole heart MRI, in which ECG triggering, gating, and navigators are no longer needed. This enables the simultaneous visualization of cardiac anatomy and function. This simple one-click solution removes some of the major hurdles in contemporary cardiac MRI that include limited ease of use and operator dependency.
Can you explain that golden angle a little bit more? And also with that, and again, probably a naive question coming from somebody that does Cartesian imaging, but how does the undersampling work in terms of, how do you describe it, I guess? Because when I think of undersampling, I think of an R factor, and then that immediately I know, okay, I'm skipping every so many lines. And in your paper, I realized that you report the number of spokes that you acquire, essentially. So how does that relate to your undersampling factor?
The golden angle is the starting point of many different implementation of radial trajectories. And the idea is that each readout or each interleaves or each group of readouts will be rotated in the K-space by the golden angle. And this particular angle allows to never acquire the very same readout over the time. So which means that no matter how long will be the acquisition, each readout will be unique in terms of the information that it brings to the reconstruction. Not only that, it also samples the K-space in quite a uniform way. That means that it's going to populate our K-space in a way that it doesn't really matter how we are going to then divide to bin this data set, that all the bins, all the different images will anyway be more or less uniformly sampled.
I may add something to the golden angle I just mentioned before, that the efficiency of how we sample or collect data of the heart is unparalleled with this technique. And so by extension of this logic, we never want to acquire the same information twice, right? So how can we cover an entire 3D space in a way that we never acquire the same information twice? And this concept is basically borrowed from nature, where petals of a flower are organized in a way that they optimize the space that's remaining, right, to grow another petal. And that's exactly the same thing. It's the golden angle that we just borrow to populate K-space in a highly efficient manner.
And when you say bin, you mean it's like a point in the cardiac cycle and the respiratory cycle in your case?
Yes, in this case, yes, because with what we call the 5-dimensional images, so images that have 3 spatial dimensions and respiratory and cardiac, that means that each single image reconstructed will be in a specific time point along the cardiac cycle and in a specific position along the respiratory cycle. So yes, each bin is one of those positions, motion resolved positions.
Can you now describe your triggers a little bit more?
Yes. So the cardiac triggers. So along the cardiac dimension, we use the cardiac triggers to define a specific temporal position within each cardiac cycle. So obviously we have a long acquisition, and we need to somehow align each single cardiac cycle in a consistent way so that we can take the information from all the cardiac cycle of that specific, let's say, the systolic the systolic phase, and put all of these information that are consistent together. And so to do that, we first extract the self-gating signal, which is a signal with a certain temporal evolution. And from that one, we want to extract just one temporal point that will tell us that that temporal point is the starting of one cardiac cycle of each of the cardiac cycles, so that we can align all of them. And this is what we are going to, in the paper, we compare it to the ECG, which is our reference in this case, which is a different approach from what is done along the respiratory dimension, which is much more widespread as a technique, because there we have a signal that we consider not in terms of temporal distance from the trigger, but we consider the amplitude evolution of the signal. So we're going to match, for instance, one extreme with the end expiration and another extreme probably with the end inspiration, and we're going to divide according to the amplitude of the signal. First of all, when I have the information from which I need to extract our cardiac and respiratory motion, I just have some what we call some SI projections, so some readouts acquired in the superior inferior direction of our field of view. And those are basically the integral of the whole signal on a single line. So it contains information coming from the whole field of view, and it is the intensity in this readout are somehow modulated for all the temporal evolutions that are happening during the acquisition. So the respiration motion, the cardiac motion, and also some other noise that are superimposed. So we need some technique to isolate each single component. So we need to isolate the respiratory motion, we need to isolate the cardiac motion, and we also need to isolate some other components that we want to clear from our information. So the principal component, as also the independent component analysis, are two techniques that do exactly that. So they take several signals that contain these information somehow mixed all together and allow you to isolate, to extract the one that pertains to the cardiac motion and the one that pertains to the respiratory motion, so that we can independently bin and then reconstruct.
What I found fascinating while he was doing that research was that the mainstay of cardiac synchronization of the MRI experiment, the EKG, actually proved to be quite unreliable in that we discovered a lot of missed triggers. And we are not even talking about high magnetic field strength, where the so-called magnetohydrodynamic effect kicks in much more and makes it more difficult, even on state-of-the-art hardware that includes the vector EKG. So it was super interesting to see, that in EKG we so much depend on these days is not that reliable, and then we have to develop better tools to address the periodicity of the cardiac motion. I think either of the techniques Lorenzo was developing may help address that need.
So what will it take to translate and use clinically?
No, I think you're right. I think the main hurdle is really trying to reduce the reconstruction time so that the data will be available as soon as possible to the clinicians. There's obviously more validation that needs to be done. I think, otherwise I don't really see any major hurdles that cannot be overcome.
How long is the reconstruction time?
Right now, the reconstruction time, so we have a dedicated computer with a lot of RAM that is able to reconstruct this 5D data set in about 5 hours.
Okay.
But again, we are engineers, we are interested in research, so we didn't put any effort into optimizing the reconstruction. So we are just trying to prove a concept that it works. And I'm sure that if industry is interested in getting that to their customers, they will be able to reduce the reconstruction time by a factor, at least 10, even more than 10. And
it anyway depends a lot on which dimensions and which resolution you are aiming for. So there's really a lot of flexibility in terms of how you use the data that you acquired and how you use them to reconstruct. So
with computational power, does reconstruction time really matter clinically?
10 years it will be reconstructed in like 5 minutes, so you just need to be patient.
Yeah, that's not bad at all. Okay, great. Then we got a lot of great stuff here. Unless there's anything else you guys wanted to talk about?
Well, perhaps. Thank you so much for doing this. It's always a pleasure to see these things, that you pick certain areas and how you do these interviews and how you approach the researchers. That's just fantastic. So please keep doing that.
Thank you. Thank you guys for participating. I always really enjoy this. I think I've personally learned a lot about different areas and different expertises. So it's been quite interesting and fun.
Yeah, thank you. Thank you.
Thank you very much.
Great to meet you guys.